and Assalamualaikum and good day to everyone and honorable audiences. I like first of all to thank the organizers of this conference and having us here. Let me start by saying a few words about my background. My name is Ain Atika and I'm a postgraduate student from Faculty of Bioengineering and Technology from Universiti Malaysia Kelantan, UMK. Before getting into the presentation, I would like to credit my supervisor, Dr. Wee Seng Kyu, who demonstrated expertise in microbiology, Dr. Muhammad Najmi bin Masri and Dr. Mahani Yusof, and also Dr. Owi Wing Tiang as in the screen for their guidance in metallurgy and material science research area. Without them today, it would be have an impossible for me to conduct this project.
So, without further, I'm very happy to present our research project entitled Effect of Shewanella oneidensis MR-1 on micelle corrosion under iron 3 reducing condition. In my presentation, I'll be focusing on three main parts, mainly on the overview of the project, results and discussion, and finally conclusion to summarize the whole project. Corrosion can be a huge topic or issue to discuss here. So we narrow down a bit the type of corrosion we are focusing, which is influenced by bacteria, also widely known as microbiologically influenced corrosion in bracket MIC. MIC can be defined as biodegradation of metallic materials influenced by respiratory and metabolic activities of microbes. MIC affects to almost industries, including oil field and marine underwater systems. At this current situation, MIC is responsible for 100 billion on the annual corrosion cost worldwide, and yep, MIC accounted between 20 to 40% within the cost. The historical background of MIC was started in March 2006. The authorities reported about 12 gallons crude oil spilled at the Prudhoe Bay of Alaska, and it was suspected pitting type of corrosion, forming holes on steel pipeline surface which had caused by the bacteria. There are a few bacterial groups reported to cause MIC. In the literature, this bacteria group were postulated to either enhance or inhibit corrosion. Just now I mentioned the incidence of Prudhoe Bay of Alaska in 2006. The researchers found the sulfate-reducing bacteria was highly damaging to MIC because they capable of enhanced corrosion by reducing sulfate to hydrogen sulfide by creating a corrosive environment. In contrast, current studies reported on contradicted findings for nitrate-reducing bacteria, as these nitrate reducers capable of accumulate nitrites as the product of nitrate. These nitrites help to develop passive iron-3 film against corrosion. Yet, current research interests started to look on iron-reducing bacteria species in genus Shewanella because these Shewanella are postulated to enhance and inhibit corrosion under different experimental settings. Because most of literatures are focusing on MIC acceleration by iron-reducing bacteria, in bracket IRB, through removal of ferric oxide on steel surface, but iron-reducing bacteria also postulated to inhibit MIC by means of transforming of insoluble iron 3 oxide to other iron 3 or 2 substances such as magnetite, hematite, and vibenite as physical barrier or protective layer within the microbial biofilm formation against corrosion under anaerobic conditions. Thanks to the facultative anaerobic of Shewanella, due to its capability to reduce a wide range of electron receptors compound such as soluble and insoluble iron 3 metals, nitrate, and nitrite. Due to this, we intend to design an experiment using the same bacteria. How this facultative Shewanella affects MIC under different iron 3 reducing conditions using soluble and insoluble iron. To investigate the effect of Shewanella oneidensis MR-1 on micelle corrosion, The Shewanella oneidensis MR-1 will be grown under either soluble iron 3 using ferric citrate or insoluble iron 3 using hydrous ferric oxide in bracket HFO. So MIC works when the metal samples incubated with Shewanella get corroded via reductive dissolution of iron 3 particles to dissolve iron 2 using either ferric citrate or HFO as terminal electron receptors. These iron 3 reduction mechanisms of Shewanella oneidensis MR-1 will remove the protective layer of iron 3 formed on metal samples, eventually lead to pitting corrosion within incubation period of time. Now we proceed to the methodology parts. We worked mainly on 3 main stages. The first stage was cultivation of Shewanella oneidensis MR-1 in Luria Bertani albi growth and broth with agitation speed of 150 rpm at 30 degrees Celsius for overnight. In the second stage, we synthesize both soluble ferric citrate and insoluble hydrous ferric oxide HFO via precipitation technique using NaO, sodium hydroxide. In this study, either soluble ferric citrate or insoluble HFO would be electron receptor for the Shewanella oneidensis MR-1. In the third stage, steel will be cut into square size, mounted to epoxy resin, and polished using sandpaper until surface mirror was obtained. Next, I would like to move to our results and discussion segment, which adds to our research project. In this segment, we have 4 different tests in their respective results. To begin with, I'll start off with pyrazine assay. The test was conducted during the corrosion incubation for a few days. Let's look at the figure 1, where the corrosion incubation took place in different solubility of ferric compounds. They have been incubated with Shewanella oneidensis MR-1. Based on the study, correction of ferrous concentration by Shewanella oneidensis MR-1 via ferric citrate reduction were approximately 4-fold greater than the abiotic control during incubation, while HFO reduction by Shewanella oneidensis MR-1, on the other hand, retained the ability to reduce HFO by almost 2-fold than abiotic control without the presence of Shewanella oneidensis MR-1. We set up a similar experiment along different solubility of microbial corrosion experiment to evaluate mycel mass loss before and after corrosion incubation. After 20 days of incubation, all corroded mycels were taken out from the vials and evaluated for their mass loss. Based on figure 2, significant mass loss of mycel treated with and without Shewanella oneidensis MR-1. In the presence of ferric citrate, Shewanella oneidensis MR-1 reduced 0.4 to 4 mm per year compared to the abiotic control, which is 0.2411 mm per year. As comparison to the incubation with HFO, Shewanella oneidensis MR-1 show lower corrosion rates, which is 0.1583 mm per year when compared to the abiotic control without inoculation of Shewanella oneidensis MR-1, which was 0.3483 mm per year. Therefore, without electron receptor, corrosion incubation with Shewanella and abiotic control was significantly lower than the presence of electron receptor. Yet, for HFO reduction, there was inconsistent data compared to the earlier pyrazine test, because mass loss of mycel was very much noticeable higher in abiotic control compared to presence of Shewanella. This data suggests that Shewanella oneidensis MR-1 was poorly direct in contact with the external ferric oxide due to higher HFO surface area. The result we obtained from the scanning electron microscopy was much more interesting because, as you can see in the image here, there are noticeable changes towards the morphology of corroded mycel with and without Shewanella. I would like to emphasize the picture of B, ferric citrate, and D, HFO, inoculated with Shewanella identified with thick layers of iron oxides formed on corroded mycel. while absence of Shewanella oneidensis MR-1 in the picture A, ferric citrate, and C, HFO, show obvious multiple crack layers corrosion morphology on the surface of micelle. To sum up, we suggest that the Shewanella indeed transformed these iron oxide corrosion products into metastable mineral, causing the Shewanella in the early mass loss testing inhibit corrosion due to the iron oxide form, which act as physical barrier against corrosion. While for incubation without Shewanella, we can assume that the accumulation of ferric iron in the vials created a very corrosive environment, especially in the presence of citrate ions, because at higher concentration of corrosive ion of citrate, it can rapidly corrode the steel surface at higher rates of corrosion. Final test was the X-ray diffraction technique, in bracket XRD. We compare iron 3 and iron 2 crystal phases present in corrosion product of iron oxides in each samples. As you can see in the figure 4, on our left, the purple spectra without Shewanella in HFO as electron receptor was abundant in vivenite and hematite compounds, but the green spectra where the incubation treated with Shewanella displayed more vivenite compounds and magnetite. For ferric citrate, the orange spectra, which is referred to abiotic control, showed no peaks compared to samples treated with Shewanella. To summarize, iron oxide treated with ferric citrates incapable to be detected by XRD detector because ferric citrate was rapidly solubilized and there was no crystallinity phases can be formed in a certain period. But the produced dissolved iron 2 from insoluble HFO in the presence of Shewanella associated simultaneously with the oxidation of steel, causing formation of other iron 3 compounds such as magnetite and vivenite by Shewanella. Okay, in conclusion, the reductive dissolution process was highly influenced by both solubility of iron 3 compounds and the presence and absence of iron-reducing bacteria, IRB. Shewanella oneidensis MR-1 is capable of accelerating steel corrosion under soluble iron 3 reducing respiratory condition about twofold than abiotic control. But for HFO reduction, Shewanella decelerated corrosion of micelle to approximately 0.119 mm per year compared to the abiotic control. Corrosion morphology and XRD analysis suggested that Shewanella utilized reductive dissolution mechanism to accelerate corrosion in soluble iron 3 in abiotic control, but in insoluble iron 3 oxides in bracket HFO, reductive dissolution of ferric iron to ferrous iron by Shewanella oneidensis MR-1 simultaneously oxidized with steel, causing another iron 3 oxide corrosion products formed such as hematite, magnetite, and vivenite, which can be acted as physical barrier against corrosion.